Okay, so you had your row independent mechanism. Our second one we need to know is row dependent. So this actually has a different terminator sequence, so it's not quite the same. What it has is that 40 to 60 base pairs that are very rich in cytosine, but they're actually poor in guanine. So this isn't a GC rich area. It's just C rich. They also have a row utilization site. So we have 40 to 60 base pairs that are rich in C, and what's called a row utilization site, for which you don't need to know that sequence, but you'll see it often named by the acronym, which is RUT. So what this looks like here, we'll kind of just keep using this similar. We have the same basic setup where you have your DNA. We don't have the big GC region, but it still forms a stem loop in this area. And so the similar concept of that we're forming a stem loop, and there is a stretch nucleotides that comes after the stem loop, but not specific with that AA's AU connections. But in addition to having the stem loop, we have this RUT site. And the way the row-dependent mechanism works is there's a row protein. Okay. I'm just going to depict it as this red circle. And what row does is it recognizes that RUT sequence. Okay? So row will actually bind to that RUT sequence. And it's the row utilization site, that RUT sequence, is conveniently always located slightly upstream, the way I drew it, from a region that, an area that RNA Pol generally pauses at. So like this stem loop region, like we showed before, where it tends to bind to RNA polymerase. The RUT sequence is always located a little upstream from a site where RNA Pol is going to pause. So with that stem loop, this binding is still occurring here, where essentially the action is that it binds, RNA polymerase binds to the stem loop and pauses. And then the row protein can come and bind to this RUT site. And then while RNA polymerase is stalled, the row protein actually runs from RUT. It scans up the whole length of the messenger RNA and knocks off RNA polymerase. It's kind of a weird concept. It's like I always picture a little engine on a train track just like hauling ass up the train tracks and knocking the train off. So you have to picture this protein having enough catalytic activity and speed that once it binds to RUT, so if we kind of put this into like a real-time two-dimensional image going on the board here, this row protein will actually speed up the RNA and knock off the RNA polymerase. So it acts like a helicase. That's the main sort of job of the row protein. So we can add that through your diagram. It's going to, the row acts as a helicase. It breaks hydrogen bonds that are between the DNA RNA complex. Because these are bound right here, right? As it's transcribing, you have messenger RNA that is undergoing hydrogen bonding with the DNA. Briefly, as it's being transcribed, Rho will break those bonds and cause the whole complex to disassociate.
So is it the Rho dependent has a GC heavy rich region that at the end of the mRNA transcript compared to the Rho independent?
Rho independent has a GC rich region.
Whoa.
Rho dependent has a C rich region. Is that what you were?
No, more towards the end of the mRNA transcript.
With the stem loop?
Yeah, where it has uracil rich region, which is it doesn't.
And that's the Rho independent, yeah, it has the uracil rich. Whenever the genes that utilize Rho, they can have different sequences in here. We often see different sequences in the stem loop, but they'd have to be complementary to each other. So you could even have, usually stem loops are full of GC just because they're so, they have such stronger bonds with the three hydrogen pairs. Are you thinking,
Well, that's what I meant with the end of that mRNA transcript, how it has more hydrogen, like 3 hydrogen bonds.
here, the very 3' end?
Yeah.
With Rho can actually break any combination of sequences. So it doesn't necessarily have to be the weaker double bond. So it doesn't necessarily have to be an AU rich region. Yeah. With the Rho dependent, yeah, because it's like a helicase, it can break apart almost any combination of sequences. Yeah. So on those types of genes, they often see all different sorts of sequences at the very 3' end of the transcript. You don't need to know the full, well, I know I kind of simplified it acting like it was a train on a train track, but Rho hitting RNA pol will actually cause a conformational change in RNA pol and cause it to fall off, but you don't need to know the full molecular details about that. Know that it acts as a helicase, it causes a conformational change in RNA pol, and overall Rho coming up, binding to the RUT and then moving up the transcript is going to actually disassociate the whole complex and stop transcription at a normal termination site. So the key points about Rho is that it acts as a helicase. So it's going to break those DNA RNA bonds. And the other thing it does is causes a conformational change in RNA polymerase. After it binds to the RUT sequence and flies up the transcript, it'll hit RNA pol, cause a conformational change, which causes this whole complex to fall apart. Transcription terminates. So these were 2 ways that I want you guys to be familiar with how bacterium can stop transcription, terminate transcription. So Melanie's question, if you, I know I've had folks say before that questions in the front you guys can't hear in the back always. So the question was, is either one of these, essentially is either one more advantageous? Is there a reason you might have one over the other? So which do you think is more energetically costly to the? Yeah, Rho dependent. Yeah, just because it takes a specialized protein, a specialized sequence to be recognized. So the only thing I have really to answer to Melanie's question is that Rho dependent is more rare. More of the genes you'll see with Rho independent termination sequences. This is more common. And it's not necessarily you have different cellular conditions, it's gene by gene. Each gene is specific. So depending on the gene and what the function is in the cell, whether you want to, and it also will depend on that 3' end of the gene, how crucial it is that you stop it exactly at a certain point. You'll use the Rho dependent more often because it's more reliable in terms of actually stopping exactly at that nucleotide. And that gets into lots of detail that's interesting, but not, not for the course. Beyond the course. If you didn't know it, if you take molecular genetics. both, right? Any other questions? Okay, so we will move to eukaryotic systems now. So that's all you gotta know for prokaryotic. So initiation in eukaryotes. So in general, we can think about differences between prokaryotic systems and eukaryotic systems, right? So this is, your general statement is in general, it's more complex. I know I sound like a parrot repeating the same thing like we did with replication, but this is more complicated, takes more proteins for the entire process of transcription. But what else? What are some major differences in terms of comparing bacteria to eukaryotes when we start thinking about transcription and translation? We start thinking about expression of genes. What are those? No operons. Yeah, no operons with eukaryotes. We can start thinking about things like RNA processing, right? Do bacteria have introns and exons? No, that's specific to eukaryotes, right? So things like RNA processing and thinking about translation, where is this happening in the cell? Where's transcription happening in the cell?
Ribosomes.
In the nucleus. What about translation? Cytoplasm. Okay, so we're starting with the basics. Transcription, nucleus, next to the DNA. RNA gets sent out of the nucleus, right? And translation happens in the cytoplasm. So you have completely different cellular compartments where some of these different events that we're talking about happen. And this will come into more detail when we start talking about gene regulation and the levels that we can regulate. If you're thinking about a bacterium, a prokaryotic cell, you can transcribe and translate the same gene simultaneously. Okay, ribosomes, this little picture we're drawing here, ribosomes are already sitting on this messenger RNA and making protein from it. Okay? In a eukaryote, you have to bundle it all up, tie a bow around it, and slap it out of the nucleus. Then do your translation and all this other stuff. So there's a lot more processing. This is a more complex, compartmentalized process. Okay. So we'll get more, as we start thinking about RNA processing in terms of gene regulation, we'll talk about that a little later. Actually, after you guys get back, probably. There's another big difference between eukaryotes, prokaryotes. So, prokaryotic DNA is arranged how? You know, I drew it short here, but where does the rest of this go? Circular, right? Is it packaged around proteins or not? No. There's no chromatin, there's no packaging of the DNA in a bacterium. It's pretty much naked DNA. So the transcription process is pretty simple. You just gotta open up a bubble and go. Okay, eukaryotes, what do their chromosomes look like?
Really bad.
Yeah, stuff depending on what stage of the cycle it's in, right? Your DNA is organized somehow around proteins, around histones. We could say organized in a chromatin. And what this means, how this relates to transcription, is you have to worry about organization of that chromatin. You have to think about unraveling it from the histone so that you can make a little transcription bubble and fit big fat proteins like RNA polymerase in there. So again, when it comes to gene regulation, right now we're just thinking about basics of transcription. But we're going to get into how do we turn on and off these genes, and the fact that your DNA is organized in a chromatin will definitely play a role here, because you have to unravel that DNA and get it unwound from all the histones to be able to access it for transcription. Does that make sense? Okay. are some major differences that will actually end up affecting how we think about transcription in a eukaryotic system. What else? We said bacteria is circular. What about the size of the genome compared to a eukaryote?
It's much bigger.
Small, right? Smaller meaning what?
The other one's bigger.
Got less junk.
Less junk. Okay, so what about, number of genes? Less or more in a prokaryote? Fewer genes, right? So in terms of everything you'd have to transcribe, you have fewer genes. So you have to think about size of the genome. If we're comparing these points, okay, bacteria have smaller, fewer genes, smaller overall size of the genome, less sequence, which means for eukaryotes, what? If you're RNA Paul trying to find a gene you want to transcribe, you might have to look for a long time, right? You're scanning that genome, and what was brought up was the junk, what we used to call junk sequence. Now we're realizing it's not so much junk sequence, but sequence that doesn't transcribe straight into a messenger RNA. There's lots of in-between interspersed nucleotides. Okay? So genes are far apart. If you're an RNA polymerase, then if we think about the size of the genome and distance between genes, so generally speaking, compared to a prokaryote, they're farther apart. So this should beg the question, how does RNA polymerase, for initiation, okay, of transcription, how does RNA polymerase find start codons? This should, in your head, it's a hard thing to visualize, but if you're thinking about millions of base pairs of DNA sequence and finding genes, you should be thinking of like a needle in a haystack type analogy here. This is pretty difficult to do. So how does RNA Paul find a start codon?
Something attaches to it. No.
Through what?
A promoter sequence.
Just tagging.
No, but that's actually a great idea. Yeah, there are areas of the genome that are regulated that way so that they can find it. Yeah, it would be great. Yeah, if you could have some kind of giant tag. Okay. But one of the ways that they handle this, these are kind of some of the complexities. I wrote this general statement. Okay, in general, initiation is more complicated, takes more proteins than what we saw in prokaryotes because it's a huge genome. Genes are spaced way far out. Eukaryotes have evolved to deal with this, okay? So they have ways to be efficient still in their transcription and not take, 24 hours to transcribe a gene that you need right now inside of the cell, okay? So some of the ways you can list out that they've evolved to handle these complexities, one of them is to actually have multiple polymerases. So there isn't just one RNA polymerase in eukaryotes. There are multiple. They aren't nearly so bad as thinking about the subunits of DNA polymerase for replication, okay? What you guys need to be familiar with is RNA Pol 1 through 3. So this is one of what we're talking about now is one of the ways that eukaryotes have evolved to handle this complexity. How does RNA Pol find the right gene? Well, what you do is you break up the work, right? You assign different genes to different RNA polymerases. So RNA Pol 3 isn't going to be running around searching for messenger, for structural genes. It has its specific job. It only transcribes certain genes. Okay, this way you divvy up the work and you can be more efficient at it. Okay. So Pol 1 does all the ribosomal genes, so all the ribosomal RNA except for 5S. So there's one exception, and that's not the 5S ribosomal subunit. Okay. 5S is handled by RNA Pol 3. Don't ask me why. I have no clue. I don't know why it evolved this way. 5S is a really important subunit though, so in some ways I guess it makes sense that you would have a separate RNA Pol for that part of the ribosomal RNA. The other types of RNA that Pol 3 handles are all of the transfer RNAs. Pol 2 does all the other stuff that we mostly spend our time thinking about. Okay, all the messenger RNA, the stuff that's going to actually be translated into proteins. Most of these, I'll just, for now, we'll just keep it simplistic and talk about some what's called small nuclear, SN, RNA. This might be a new type of RNA for you guys, maybe not for some who've heard of this before. Small nuclear, snRNA, and we will talk about what those do and what they are when we start talking about RNA interference and RNA silencing and stuff to do with eukaryotic gene regulation. It turns out there's a ton of RNAs that run around in our cell and do crazy things that they're discovering more and more recently. So there are other functions for these polymerases as well. But for you guys, just focus on RNA Pol, does all the ribosomal except for 5S. Pol 2 does all of our messenger or fun, exciting stuff that gets translated into proteins. Some small nuclear. Pol 3 does 5S and the tRNA. Okay. So, reason, or I guess one thing that eukaryotes have done to deal with this complexity is to evolve a bunch of RNA polymerases. Okay. Another thing is they assemble a bunch of what's called general transcription factors. These are proteins. So for initiation transcription in eukaryotes requires the assembly of what we call general transcription factors, and these are proteins. But these, and you'll see, because biologists love acronyms, GTF. Okay, so GTF stands for General Transcription Factors, if you ever see that. This assembly of all these proteins, these transcription factors, has to happen before RNA polymerase can sit down and start its work. Okay? This assembly is actually necessary for the function of RNA polymerase, especially RNA Pol 2. So you have to gather all these other proteins together to actually have this whole system work. And we'll describe those GTFs, these general transcription factors, here in a minute. Okay. But this big concept here is to deal with the fact that we have a giant genome, we're trying to find start codons in the middle of this haystack. By doing that, we make it more efficient by having multiple RNA polymerases that are for specific jobs, and they tend to hang around in certain parts of the genome where they're needed. And then you also have to have this assembly, and this ensures that everybody's in the right place, that you didn't accidentally find what you think is a start codon but isn't actually the needle in that haystack. To help ensure the process is accurate, this whole assembly of other helpers is needed. To make this happen, and they are going to help find that needle in a haystack as well. So it's not just RNA polymerase doing this job all by itself. Okay. That's the kind of the general concept behind why would you add in all these other players, all these other proteins to the process. So next piece, we're okay with this? Okay. next piece is for initiation, we have to think about where that start codon is located in terms of what's the promoter sequence, okay? Because we talked about promoter sequences in prokaryotes. So eukaryotes, say if we have a stretch of DNA, right? Promoters are much more variable and longer sequences in eukaryotes. We are just, for right now, we're just going to think about structural genes. Promoter sequences of like the ribosomal and tRNA genes, they're a little more complex. So we're just thinking about how we get a messenger RNA. So those are structural genes, okay, in terms of promoter sequences. What do you guys remember from before? What's a common eukaryotic, what comes to mind for promoter sequences for eukaryotes?
Tata box.
Yeah, Omar said it. So the Tata box, right? So you have a TA, T, and then a string of A's, right? And you'll see this called the Tata box. This is not always, but usually around -25. Okay, so 25 base pairs. To designate a string, the type of pyrimidine, so whether it's cytosine or thymine, that varies from gene to gene and species to species and all of that kind of thing. So this again is that consensus sequence for a promoter. Okay, the most common, the most average sequence that we see is an adenine. Sorry, I missed a cytosine. I got excited about pyrimidine. So an adenine, upstream of that is always a cytosine, and then our two pyrimidines. Downstream of the CA is 5 pyrimidines. This is a common sequence that we see with variations. Okay, and then so the Tata box plus that consensus sequence is what, if you were referring to your core promoter, that's what you're talking about. Make sure I have enough room here. So we're going to draw so this whole, any questions about our promoter sequences, what this looks like? So what it looks like for initiation, piling all these GTFs, we're going to draw all the GTFs bound up there to the promoter sequence. Okay, how we start this whole transcription process in eukaryotes. It's going to look somewhat similar to prokaryotes in that you have a little transcription bubble and all these proteins come in and sit down on the DNA. Okay, so also similar to thinking about bacterium. So what subunit on that we just talked about with RNA polymerase for prokaryotes, what subunit recognizes DNA and binds to the promoter? Yeah, Jason's right. So sigma is the key one who's like, hey, there's the needle in the haystack, there's my gene start site, there's a promoter, I'm going to sit down. Eukaryotes are the same in that RNA pol 2 doesn't recognize and see the promoter sequence by itself. It needs help. Okay? It needs additional proteins to help find that start site. Okay? So you add your DNA up here, and you have your Tata box. Okay? Think about that being our core kind of promoter sequence. Okay, we have a bunch of general transcription factors, these GTFs that I'm going to draw up on the board, and hang with me because there's a bunch of them, okay? So a bunch of these general transcription factors are what's going to bind, and they're separate from RNA polymerase. So this is a key difference too from eukaryotes, where is sigma a separate protein or part of RNA polymerase? Part of it, it's a subunit of it. With eukaryotes, these are separate proteins. They work together and they physically contact RNA polymerase, but they are a separate protein. Okay, so we have a bunch of general transcription factors, and they're all named. I'm going to put this over here. TF for transcription factor. Okay. All of them have TF in their name and the number 2 because they help out who? RNA pol 2. Okay, so transcription factor 2, and then the next thing is what will change. So there's TF2, and there's a bunch of letters. So there's D, B, F, there's H, E. 1, 2, 3, 4, 5. We have 5. Okay, but all of them have the same beginning part of their name, TF2, and then D, B, F, H, and E. So 5 total. You can do that. Compartmentalize your studying. Okay, 5 letters up here. TF2B, okay, is the one that's going to sit down and recognize this Tata box. Okay. So TF2D, and TF2D is unique because it has a subunit on it that's called TBP. TBP stands for Tata binding protein. This is going to start to sound really redundant. Okay? So TF2D, one of the subunits, what I'm showing by this big purple box is like this is one big protein, and they're separate subunit parts on it that do a specific job. The Tata binding protein, turns out it actually needs help. So it has other subunits to it. Okay? There are associated factors. They're known as TBP associated factors. So the acronym is TAF. So these TAFs. So you have 2 TAF subunits that help this Tata binding protein. These are all part of TF2D, which is one of the transcription factor proteins that helps RNA pol 2 eventually get up here on the board and bind to DNA.